DNA, it stands for deoxyribonucleic acid. It's very simply put a biopolymer, so it means it's just a string of subunits or components arranged in a particular order. We call them nucleotides. They come in 4 different flavors. We simplify it by calling them A, C, T, and G. Each of our chromosomal sets is made up about 3 billion of those nucleotides, and they're arranged in a specific order to confer all the information that is needed by each of our cells. And each one of our cells in any given person has the exact same DNA in every one of their cells. The arrangement of the DNA is composed of what we call genes. Those are the sort of the substructure of the genome. We think the best guesstimate now from the genome efforts are that there's probably about 25,000 genes. It's just what happens after the genes, they get processed and give us the diversity that makes us what we are. There are a series of diseases that we know differ depending on whether or not you inherit them from your mother or your father. If we think of your 23 chromosome pairs as your instruction manual, and that's the manual of how you make a person, that sometimes that manual makes mistakes. Cystic fibrosis is the absence of a single gene out of those 25,000, and it manifests in a bunch of different ways. It's a very systemic disorder. It affects just about every organ system in the body. The pancreas is an organ that's involved in digesting your food. So in CF, the ductwork in that pancreas, it sort of looks like a bunch of grapes, and you think of the stems as the ductwork, that those ducts become blocked and the enzymes can't make it to the intestines, and so there's nothing to break down the food, so it goes right on through. This same process that's going on in the pancreatic ducts is also going on in the airway. So when this gene is missing, it prevents the cells from being able to move ions back and forth across them. In CF, the ion channels are not working appropriately, so the salt doesn't move, and as a result, the lining of the airways becomes dehydrated, so it doesn't have the fluid it needs. So all the mucus that's in there that's normally supposed to capture the bacteria aren't removed, and the immune system just keeps coming and coming and coming. And as the end result is that the immune system ends up tearing up a lot of the healthy tissue. CF, as an example, is one of the more common diseases, and even though it's common, it still only affects about 1 in 3,000 people. It is a recessive disease. This is an ion channel that affects a number of tissues. And if you think about this from those people who are completely unaffected, they have a certain number of these ion or chloride channels in their cells, and everything works just peachy keen, fine and dandy. But if you are a parent of a CF child, and maybe even a brother or sister of a CF person, there's a likelihood that you would have only half the number you need. But that half is still enough to keep you completely unaffected, and we call that a carrier. But because half is enough, you're unaffected and only have the carrier status. But if you're missing both copies, both from mom and dad, you don't have any, and that causes the disease state. We have some kids who have relatively mild disease, and CF is never mild, but some is milder than others. But we also have those who are very, very severely affected. So what we've done is we've gone around the country with my colleagues down at the University of North Carolina and we've gone around the country and collected clinical records, so the doctor's records of how these kids are doing, and we've collected blood samples on them, so we have a DNA source from each of these kids. And we've compiled these and looked at them to see how their genetic makeup compares. So the process that we've taken with these patient collections is to go through and look at every chromosome and see what kind of variation they have in each of their 25,000 genes, and try to compare those variants with how well or how poorly they do clinically. The end game here is to try to figure out a better way to treat this disease. So we've somewhat exhausted the existing therapies. So if we find that a particular gene associates with the disease severity, it must have some functional effect on that gene. We have an incredibly large arsenal of drugs out there between the different pharmaceutical companies and research institutions And now we're in the phase of trying to match those up. So we find that gene A is very, very important for the disease, and its protein can be changed by a certain set of drugs. We can now try those drugs and see if they will mimic the effect that Mother Nature has accomplished. So if losing an enzyme's activity is a good thing in CF, then we can use a drug that might inhibit that enzyme as well and try to duplicate the natural variation effect. Metabolism is a big issue in CF, so we're doing metabolic and nutritional studies, and things that have been done at the basic level like that in our laboratories are now getting moved forward into the clinics in what we call translational medicine. So now those discoveries and things that we found in our cell lines, in our mice, and so on and so forth, we now can test those out in patients. And so we also have a large number of clinical trials where the drugs can be tested to see how well they work in patients. But we're finding from the genetics is that the reason that some people respond differently to a drug than others is that they may have enzymes that metabolize that drug differently, so one person may need more than the other. So if we can go in and understand our DNA better, we have the ability then to figure out the right amounts or the right drugs to use beforehand, rather than having to do the trial and error approach. And this kind of mentality is what we refer to as personalized medicine, that if we can make the way we treat diseases a bit smarter than what we do now,